Thank you for attending. My name is Thomas Farb, CEO and co-founder of Thrive Bioscience, a private company based in the Boston area. And in 2014, we started Thrive to automate a neglected but critically important area of life sciences, which is cell culture and stem cell culture. At the bottom of the slide, you see the first cell line factory in 1952 at Tuskegee Institute, producing HeLa cells. Now, I'm sad to say that there's been limited innovation since then. Cell culture is done pretty much the way it was 65 years ago. And even though cell culture is at the root of most biomedical research, there have been little improvements, and it leads to waste of billions of pounds and much time bringing medicines to patients. Our objective is to produce instruments and software tools that will produce consistently healthy cells, and for the first time with data. Or as we like to say, good science needs good cells. So a few key points about Thrive. There are few solutions from existing vendors, even though it's a very large market. And we've solved many of the key problems that exist in cell culture, which involves imaging live cells, analyzing them, and manipulating them. We have a family of products ranging from about 70,000 to almost 700,000, with increasing degrees of automation. And they don't require FDA approval. We project having over $100 million in revenue in 5 years, and with a significant amount of recurring revenue. We've amassed a portfolio of 42 patent applications, 8 of which have issued, in large part because this instrument has never been developed before. And we have extensive collaborations with the Broad Institute and with the Harvard Stem Cell Institute. In fact, each has contractually committed to purchasing a beta instrument and 5 production instruments of our most expensive. They're each using it for good examples of where cell culture is difficult. So the Broad found that after 5 to 6 weeks of the stress of cell culture, there was enough genomic drift that it was affecting their research. And the Harvard Stem Cell Institute is using it for the difficult task of growing iPSCs. On the financing side, we've raised $18 million from 7 angel groups in the United States, from family offices such as the Estee Lauder family, Parker Brother family, which makes Monopoly, so we're spending Monopoly money, and a $7 million Series A. We're in the process of starting raising our Series B, targeting $12 million at the beginning of 2019, and mostly to fund the commercialization and the launch of our first product in June of next year. So what is cell culture? So we're actually trying to create in the lab, in vivo, the environment that cells live in, and we do a very poor job at it. The way cells are actually kept in the lab bear little resemblance to our bodies. So cells are kept in these dumb incubators that only control gases and temperature and not important things like reagents or pH. But they do have a very sophisticated humidity system which consists of a pan of water. In order to even look at these cells, we have to take them out, take them over across different environments to a microscope or to a hood to perform experiments, leading to contamination, cell stress. As an article in Drug Discovery World points out, cells need very fine-tuned environments to survive, and current incubators do not offer this. Current cell culture leads to inconsistent results and undocumented results. When an experiment doesn't work, we don't know why. And I would say that when an experiment does work, we can't go back and look at why. It is not a well-controlled process. There's genomic drift from the stress of multiple environments. There's significant mislabeling and contamination. This is an incredible statistic. NIH found that 10 to 20% of all cell lines that they surveyed were mislabeled. Mislabeled means that a researcher thinks they're studying pancreatic cells, but might be studying hepatic cells. And contamination rates actually lead to not only contamination from things like mycoplasma, but DNA that crosses across cell lines. So basically, cells are increasingly distant from the cell model, which is us, the patient. We clearly need better data and better biology. I'd like to give you just a quick case study on cell culture. So I have the sequel to Goldfinger, and it's about do you want your cells shaken or stirred? So this is a study reported in Cell in 2016, and a set of data that should have taken months took years. And the reason is that the two groups were doing what they thought were the exact same processes, the same reagents, the same cell lines, and we're getting different results. So it was Lawrence Berkeley National Labs and Dana-Farber. And for over a year, they kept running these experiments over and over again and getting different results. And because they were under one grant, they actually had to reconcile their differences. 2/3 of the time, irreproducible results are not reconciled. It turned out that one of them was shaking their cells and another was stirring their cells. This is one of 100 different parts of the process of cell culture, and yet these variations have very significant differences on research. So our mission is basically to correct that, to provide instruments and software tools that'll give us healthy cells consistently and for the first time with data, and thus significantly improve reproducibility, sterility, scalability, and with documentation. We have a full line of instruments that we're producing, instruments, software, protocol libraries, for the first time a major database in cell biology as well. On the instrument side, we have 4 instruments, a family. On the low end, we have the Cell Assist, which is about $70,000. It's currently in beta test. And it basically sits next to the incubator, doesn't replace the incubator, it improves the incubator. And it improves our judgment. It provides reports and guidance while building a database. On the upper end, we have the Alpaca cell culture system, which is about $650,000. Takes the same software and imaging that is done in the Cell Assist, wraps it in a controlled environment, and adds robotics and fluidics. That's an alpha test with the Broad Institute. So let me talk a little bit about Cell Assist. So currently, researcher takes the cells out of an incubator, spends several minutes looking at them under a microscope, returns them to the incubator, and guess what? All the data and images are lost. There are no laboratory information management systems in cell culture labs. Instead, with the Cell Assist, they place a plate of cells into the Cell Assist, and in less than a minute, we take 250 images. If you want more images, in about 3 minutes, we take 1,000. And we analyze them and produce reports and guidance as to what are the next steps that need to be made. And for the first time, we're building a database, not only of images of the cells, but of the reagents and of the processes. So that when an experiment doesn't work, you can actually go back and look at what the problem was. Or if it does work, you can reproduce those results. It's in beta test at some leading institutions, Harvard Stem Cell Institute, Broad Institute, Mass General Hospital, Stanford University Stem Cell Core, Boston Children's Hospital, Keio University School of Medicine, and we just added University of British Columbia in Canada. On the high end, we have the Alpaca, which we call cell culture in a box. That wraps the Cell Assist software and smarts into the controlled closed environment. We have in there incubation, refrigeration, 3 microscopes, fluidics, robotics. A small example of one of many innovations, you see in the middle of the instrument a stack. That is our incubator, and we call it a cell hotel, and each of the cell plates has its own isolated, contamination-resistant hotel room. Very different from today's incubator where you reach in and pull cells out. So why would somebody spend $70,000 to $650,000 on such an instrument? Well, it leverages researchers by a factor of 2 to 5 times. Paybacks are less than 12 months, but more importantly, it does things that you can't do currently. The controlled environment reduces contamination and genomic drift. The onboard sensors control the environment and modify it. The image processing basically makes the judgments rather than a person and does it consistently, and does this with documentation. So our products and our IP don't just cover cell culture. Tissue culture, 3D cell culture, pathogen culture all suffer from the same lack of well-controlled processes and lack of data and lack of documentation. And we're actually not just culturing, we're also analyzing the cells with microscopes, 3D imaging, and other things. We're engineering the cells, such as with iPSCs, and we're producing these cells. I mentioned the market size is over $18 billion per year. Probably more importantly, about 3/4 of that market is commercial. Biotech, pharmaceutical, cosmetics. One of the reasons the Estee Lauder family is an investor. Cosmetics are one of the largest users of in vitro toxicology. Other applications include assay validation, building of cell libraries, cell screening. We have a team of 15 people that focus mostly on the software and prototyping of instruments. We use an outside engineering firm for production engineering and manufacturing. My own background, I've started over 10 companies, including H&C Software and Exact Sciences. I was in the early management at Cytic, where we automated cytology labs, quite a good comparable. Cytology labs were also ignored and unautomated. Cytic ended up selling for $6 billion, 10 times trailing revenues to Hologic. Another comparable is Ventana Medical Systems, bought for $3 billion by Roche. They automated pathology labs. We're automating much larger and non-FDA regulated cell culture labs. My co-founder, Alan Blanchard, is responsible for many of the major inventions in genomics. The chairman of our board, Guy Broadbent, ran the largest laboratory products group in the world at Thermo Fisher. And we also have on our board Brock Grieve, who's executive director of the Harvard Stem Cell Institute. Growing of iPSCs has a lot of pain points and is one of our major applications. So we've raised $18 billion to date, a $4 million seed round in 2015, $7 million Series A in 2016, a convertible note in 2017 and 18, and early next year we'll be raising our Series B to basically fund our commercialization of our first product, the Cell Assist. So I'd like to end with a couple of quotes from customers. We have a complete lack of tools to help us in this increasingly important task of cell culture. And cell culture may be the only remaining island of outdated manual processes surrounded by an ocean of automation. We're basically bringing analytics, automation, better biology to the life sciences. We're interested in finding investors, customers, collaborators. I'd be glad to take some questions.